For the introduction. Good morning, everybody. My name is De-Fen Mou. I'm really glad to be here to talk about what we are doing in our lab. I started this position last year in January, so it's been a year and a half. So I feel like I'm doing the interview again, kind of starting nervous. So thank you, everybody, for coming. And here, our lab name is Agricultural Vector Entomology Lab. So how it started is this. We had empty lab. We didn't have anything except the benches and some chairs. And now we have a lot of stuff going in here. So you can also see Laura is here. So today we are going to present what we are doing in the lab, and also some of our lab members is going to present. Before we jump into the study we are doing, I'll give a brief introduction of disease triangle. Maybe you've seen this before. It's Plant Pathology 101. So for a disease to develop, you have three important factors. Plants, let me do this. You have to have the host plants, the pathogens, and also the favorable environmental condition. So when you have these three factors, then there is a possibility for a disease to develop. But for some of disease, for some pathogens, they need insects to transmit them. For example, some viruses and phytoplasmas, they are colonizing in plant phloem set. So when insect feeding on this phloem, they can uptake these pathogens, and then when they move around, that's where they can transmit the virus or phytoplasmas. So for these pathogens, insect vectors, they play a key role in the disease development. So without insect, then this disease will not transmit and hence no disease. So this is illustration of insects feeding on a plant. So this is aphids. They have this long, slender stylets. They can go all the way into phloem set and suck up this phloem and then sugar water from the plants. And then if the plant is infected with any pathogens, and then the pathogen can go into their body. So you can see here, this is illustration of a plant hopper. Some of the pathogens, they only stick to their stylets, and then they can transmit by that way. But some of the pathogens, they go to all the way into their body, enter the foregut, midgut, hindgut, and then they can also come out from the gut, past the gut barrier, and circulate in the hemocoel. And then these pathogens, they can also go to the salivary glands. And when these insects fly or jump between plants and then feed on the healthy plants, then the pathogen get to transmit it. So this is a good illustration. You can see how the insects can transmit plant pathogens. So based on this, I have this new disease triangle for those pathogens transmitted by insects. So here, the key component, insect pests and vectors, pathogens and plants. So this is kind of our major research interest. And for the plants, we are focusing on the plants and crops that are important in Everglades agriculture area. So for here, we are a big winter vegetable producers, and we have a biggest sugarcane growing area, and rice is also important as a rotational crops. What we are studying is insects and pathogens associated to these plants. For example, thrips, aphids, and leafhopper and plant hoppers, they are all very efficient in pathogen transmitting, and these pathogens are occurred in these plants. So within this system, what we are interested in is the factors that can affect these insects directly. For example, their life history, their population dynamics, ecology, or insect behaviors, and they might also have insecticide resistance and their host range. All of these factors can directly affect this disease triangle, and within this, we are also interested in the interactions among these vectors. For example, insects can transmit the pathogens, but how they do it. And also, what is the response of the pathogens once they infect these insect vectors? They can change the insect's behavior or their physiology or their preference. And between the insects and plants, they also interact in terms of plant defense or insect feeding on the plants, and then it triggers their immune response, but they have the counter immune suppression. So we are also interested in these interactions. And our goal is to use this knowledge to help growers to protect their crops from the disease and vector problems. So first project we are working on is the lettuce thrips, the INSV system. So here, as I mentioned, the lettuce is a very important crops. And thrips is one of the common pests occurs in lettuce field. And the impatiens necrotic spot virus is a virus that can be transmitted by thrips. So we have this triangle here for this project. For more details, so lettuce is an important crop in Everglades agriculture area. Florida is the third largest lettuce producing after California and Arizona. Because we have a good weather, so during winter time, that's when we grow lettuce. Annually, we have 11,700 acres harvested in this area. And INSV is a virus that was considered a pathogen of ornamental plants. But in 2006, they found INSV in lettuce field in California. And since then, they had some outbreaks here and there. And until 2019, they had a huge outbreaks, and then they experienced a total loss in California lettuce production. And this virus was also reported in Arizona in 2021. This is the symptoms of INSV infection in lettuce. So you can see some of the discoloration, necrotic spot, and stunt growth here. So any stage and any cultivar of lettuce can be infected by INSV, and the symptoms may vary. So we need a lab-based diagnostics to accurately identify if this is INSV infection. And INSV has more than 300 plant hosts. So all lettuce type can be infected, and also some other vegetables include lettuce, celery, spinach, and basil, and even pepper can be susceptible to INSV. You can see some of the symptoms here. This is the celery infected with INSV. And this virus is only transmitted by thrips, and they are not going to transmit it by equipment, tools, or like human activities. So thrips is very important, as I mentioned, is a key component for this type of a virus transmission. And Western flower thrips is the major vectors of this INSV. So just to sum up the current state of INSV in Florida, this virus was reported in Florida, but in ornamental plants. So we don't know if any of the lettuce are infected with this virus or any other vegetables we grow here. And the thrips, they're present in Florida, but what we don't know is how many of them are there and what is the species composition, because it's not every species that can transmit the virus, right? So we want to know if the major vector, the Frankenella occidentalis is the major species in lettuce field or not. And based on this, what we are doing it here, the mission here is we are monitoring the thrips population abundance and species composition in lettuce, and Yavan will going to tell you more about this.
Thank you. Good morning, everybody. My name is Yvonne Williams. I'm the biological scientist for the Vector Entomology Lab, and I've been here since February of last year, so pretty much when the lab was just tables and chairs. So I'm going to be summarizing the project that I've been working on, which is monitoring thrips for abundance and species composition in lettuce. So an important aspect of finding vector transmitted diseases is finding the vector itself. So one of the first challenges we had was how do we efficiently sample these thrips? As you can see here, thrips are very small. So manual scouting can be often labor intensive and very difficult. So our main objectives were to find a way to efficiently sample thrips and also to monitor for the vector. So we used 3 sampling methods based on various studies that proved to be efficient. The first was pan traps, colored bowls filled with solution for trapping and preserving insects. The second were sticky traps, double-sided cards with adhesive to trap dispersing insects. And the third was whole plant sampling, where we harvested the entire plant and placed it in an ethanol solution so that we could collect any insects that were present. For the pan traps and sticky traps, we used blue and yellow varieties because these were shown to be effective in attracting thrips. We had 2 survey areas. The first was on the EREC research farm. The second was on a commercial farm. For each survey method, we had 3 reps of each trap, and the samples were weekly. After the samples were collected, they were brought back to the lab where they were further processed. So we processed them and separated the debris and the bycatch from the thrips, and then we counted the thrips on the sticky traps. So in the first 4 weeks of our collection for the EREC farm, these were the results that we got. So the blue sticky traps we found had a higher yield of average of thrips, and we found a significant difference of weeks 5 and 6 after the planting of the lettuce. But overall, the results proved to be sampling and date dependent. We're still working through the samples so that we can have an overall picture of the efficiency of each of these traps, and also so that we can figure out the species compositions of the samples we collect. So now I'm going to hand over the presentation to Tennyson, and he's going to talk to you more about sampling for INSV and lettuce, and also host resistance for thrips.
Thank you, Yvonne. All right, so once again, my name is Tennyson Birinkinyinkoma, and I'm the new master student in this lab. And I'm going to present today my research, which is titled Improving Management and Resilience to Thrips in Lettuce in Florida. So my research has been divided into 3 objectives. The first objective is to monitor infectious necrotic spot virus in different lettuce farms in Everglades agricultural area. And the second objective is to monitor thrips population in the field. While the third objective is to evaluate lettuce germplasm resistance to Frankliniana oxyentaris, which is the vector. So today I'm going to present only the first and the third objective. So on the first objective, with the help of lab team, I've been collecting 60 lettuce samples from 6 different farms that are located across the Everglades agricultural area. In collecting these samples, the main priority has been to look for plants that have symptoms of some pathogen attack. And in case that some farms don't have plants with a lot of symptoms, I'm looking for randomly looking for plants that can be selected because the virus is systemic. And that means it can also still be inside the plant while the symptoms have not been shown yet. So after collecting these samples, I'm doing molecular disease diagnostic using reverse transcription assay, using PCR, and the results are going to be shared later. I'm not going to share today. Moving to the third objective, which is about looking if we have some kind of resistance within the lettuce germplasm. Before going there, I want to explain more about what is life cycle of the thrip and how this life cycle is also related to thrips, to virus acquisition and also inoculation. So the cycle starts with ovipositioning, which can be done on plant tissue that is on leaf, stem, or flower, and it can take 3 to 4 days for that egg to transition to first instar larva. And this first instar larva can acquire the virus after feeding on already infected plant. And it can take 2 days for the first insect larva to actually transition to the second insect larva, which can also acquire the virus after feeding on the already affected plant. It also takes a combination of 2 to 3 days for the second insect larva to transition to the pupa stage. So the pupa stage and pupa stage, they don't actually acquire the virus, even if they feed on the already affected plant. The other important stage is the other stage, which can take like 4 to 5 days for the pupa stage to transition to this other stage. And this stage is the one that transmit the virus throughout their lifespan when they feed on non-affected plants. It's also important to mention that the other stage will not transmit the virus to the eggs. So the virus acquisition is only done in the first insect larva and also the second insect larva. So when looking at resistance of plants to insects, we're focusing on the adult. We are focusing on the other stage to see if this other stage can oviposition or if it can also multiply within a certain plant. So we understand, we know that plants are different from other organisms like people or animals. When they are being attacked, they will not run away. So they have to use a different kind of mechanism, which I'm going to explain now. The first mechanism is called antibiosis. This is explained in a way that the presence of chemicals in the plant, which are actually called plant secondary metabolites, can make the thrip or the insect die or not multiply after feeding on it. So to understand this one, I'm doing the no choice assay, which is involving 41 lettuce cultivars that are going to be evaluated for thrip survival, fecundity, and also feeding damage. These cultivars involve wild lettuce cultivars and also breeding lines that are actually used by plant breeders to look for plants that have desired characteristics. And I'm also using some cultivars that have already documented resistance to other arthropod pests to see if these cultivars can also be resistant to the thrips, and also some other cultivars that have great economic relevance to the Everglades agricultural area, which can be good news if we find some resistant cultivars because we can easily recommend to the growers. So this is the illustration of how no choice assay is being conducted. I'm making a cage and put only one cultivar in that cage and put 7 thrips inside to see if these thrips will not have, so these thrips will not have any choice to, but also only feed on this plant. And we see after 14 days if they have survived or if they have multiplied. In real life, this is how these cages are. I've made them using modified dairy containers. The other mechanism that plants have for resistance is called anticyanosis. This is actually the ability of the plant to attract the insects, which can be done due to the leaf coloration or the presence of other physical characteristics like presence of trichomes, which means that if plants have a lot of trichomes, they can affect the plants, the insects that can feed on them, because the insect will not be able to reach to the plant tissue. Or if it does not have trichomes, that means insects will easily feed on them. So on this one, I'm giving the insects the choice. That's why it's called choice assay. I'm giving them the choice by giving them 10 different cultivars and put 200 thrips on the middle. And after 24 hours, I'm going to see if which cultivars has a lot of thrips or has a lot of damage, because that means that cultivars is more susceptible, while the one that has less thrips and less damage is more resistant. Thank you so much.
Okay, thank you, Yavan and Tennyson's presentation. So the next project is invasive thrips on vegetable crops. So in this system, we have this thrips parvis spinus is a new invasive thrips in Florida. and they caused a lot of damages on peppers. So for system, we don't have any pathogen components yet. We don't know if they are transmitting any virus, but now because they cause a lot of direct damages on pepper, so we are focusing on this one and then see if we can help growers to mitigate the impacts of these thrips. Invasive thrips in United States is causing a lot of problems, especially in Florida because our location and the warm weather and a lot of human activities. We found a lot of invasive insects here. And for example, the chili thrips, they were found in 2005, and then now it's a major pest in strawberry and some other crops. And Asian bean thrips, it was found in 2020, and then it's also causing a lot of economic damages to beans. And now the Thrips parvispinus is a new invasive species and a potential threat to our crops. So thrips can cause direct damages to crops by feeding and oviposition. And also, as we mentioned, they can transmit viruses, some of them. So that can be bigger problems. This thrips, parvispinus female, you can see the photo here on the slide. And then they hide on the in the pepper flowers like this. So it was firstly detected in Orange County in 2020. And then 2022 and 23 growing season, they caused huge damages on pepper crops. So they almost experienced 100% total loss. So it's a big problems. And now these thrips, they were detected not only in Florida, but also Georgia and potentially Pennsylvania and North Carolina and these states. So some of these places, they are hubs for many vegetable crops, so they can cause even bigger damages in the future. So what we are doing now is try to develop a method to mitigate the impacts of Thrips parvispinus. And so our mission here is firstly accurate species identification, because thrips, they are very small. So we bring the samples into lab and we identify them under microscope. And we also collaborate with FDEX in Gainesville, so they help us ID them accurately. And we are monitoring the thrips population in the field, so they can so the growers, they can have a better informed management decisions. And we also try to see what is their host range, because these are polyphagous insects. They can feed on peppers, but also other crops like cucumbers, beans, or squash, eggplants, many plants. So we want to see what is their natural host range. And we are collaborating with Dr. Julian's lab and Anna. We're also conducting insecticide efficacy trial. And future, like in soon, so in the near future, we're going to do some host preference experiments in the lab and then see if there's any natural host plant resistant to against these thrips. So with that, we'll have Laura to tell you a little more about this.
Okay, thank you so much for the introduction. I'm Laura. I'm agronomic engineer, so I'm from Colombia, and I'm going to explain you this part and my contribution to this project. So first, we were trying to understand Thrips parvispinus behavior. So that's why we were first understand how they were growing. So we simulate these growing conditions in the insectary. So as you can see here in this picture is how we set up our plants. So we were growing them from seeds, and then we have different growth stages. So we were using these plants over here to feed our colonies. Our colonies were in these cages, so we were feeding them according, so with the frequency that they were feeding on the plants. Other objective of this project was keep the colony to use the trips for further laboratory and greenhouses experiments. So the second project that I've been involved is the pepper field sample collection. So with Anna's help, we were taking samples from 2 different locations, and then we were evaluating the sampling methods and monitor methods. And then also we were identify the trips that were in these 2 locations. So here, I'm going to show you the 2 locations, and then later on, I will going to explain you the results that we got from these 2 locations. So we choose these 2 locations because the locations were previously reported as infested fields. So for our identification, we were using these main characteristics. So one of the main characteristics from these trips parvispinus were the forewings brown, this side, and then with a base sharply pale, this side. Also, we were looking for this yellow antennal segment in the third, and then basal half in the fourth, and 5 that were yellow color. Also the legs, and then here in the pronotum, this characteristic sera from these strips parvispinus. So I'm going to show you now some of the preliminary results that we got from these sampling methods. So here in this part, you can see how we set up the pen traps. As my partner was mentioned before, we were using these colors because we're reported before as methods to monitor trips. So here, the results. So in the x-axis, I have month, and then the y-axis, I have, in this case, trips parvispinals, and in this case, my unit will be individuals. So for the first location, we have like a stable trend. So we can see like no more than 0 or 1 at least. And in the second location, here we can see in the month of March that we have more than 50 individuals. So we were analyzing like why this was trend was happening. So, and then the first idea that we have is because this trend is because some harvest practices. So, but we're still working on to interpret these results. So here, I'm going to show you the other trips species. So in the first location, the same, we have like a stable trend, but in this case, we have a range from 0 to 100. And in the second location, we also have in this month, this unique trend. So also we were discussing and thinking that it's because the harvest practices. Here in this figure, I'm going to show you average trips parvispinus. In this case, we were collecting leaf samples. So, and then we were analyzing whole cycle. So we're analyzing vegetative stages and also flower stages. So here, the green dark color represent the bottom leaf. The light green color represent the top leaf, and then this orange color represent the flowers. So here again, we have our months, and then we have individuals per plant sample. So in each month, you can see like the 3 different colors. Here we don't have bottom leaf because the plants were too small. So, and then we have different growth stages also. So, but as I was presenting before, we have a stable trend from 0 to 1. So we don't have more than that numbers. But here again, in the second location, we have higher numbers in this month. Um, the interesting part in these samples is that we have higher numbers on the bottom and top leaves. So we are trying to understand why that happened because we don't know exactly the behavior of these thrips. So the main conclusion that we can have from this graph in comparison also with, with this other graph that is showing the other thrips species. As you can see here again, we have higher numbers, but in this case, the thrips were on the flowers. So we're still trying to understand why they were in these parts, like in bottom leaf or top leaf. So our conclusion until like this preliminary result is like we have to learn more about their behavior. We don't know like how to interpret this from now because we have a lot of factors involved. So we have the grower practices, trips varispinus per se behavior, and so we're still working on that and then to give more accurate information to the growers. Thank you so much.
Thank you, Laura. So the third project we are working on is rice plant hoppers and RHBV. It's rice hoja blanca virus system. So here, this is similar to what I presented about the INSV, lettuce, and thrips. So we have the plant hopper. It's present in Florida rice field, and we have the rice, but we don't know if we have this virus or not. So for this system, so rice is an important crops in Everglades agriculture area. The rice is used as a rotational crops for the field. And annually, we have approximately 23,000 acres planted in this area. And flooding during the rice growing season is very helpful for reduce soil insect pest pressure, and also it's good for the soil tilth. So it is beneficial crops, not only the as a cash crop, but it's also good for the environment condition. And for this rice, these plant hoppers, it's not, we don't have it here, but they are in Asia and Europe and Africa, many other countries in rice growing countries. They can be huge problems for rice production. So when we are monitoring insects in the rice field, this is also one of our objectives to see if we have any of these exotic plant hoppers in the field. Then if we detect them before they are causing a big problem, before their outbreaks, then we'll try to let the growers know and then FDEX know so we can prevent them become a bigger problem or establishing their population. So these plant hoppers, they can feed on the plants, and then as the same, they can feed on the phloem set, so it can cause problems on the rice. So this is what they can cause on the rice. When the population level is high, they can cause so-called hopper burn. So you can see here, So you can see here the yellow thing here, it's not normal rice growing field, right? So this is because the hoppers, they're the number of them are too high, and then they feed on the rice, and then this rice cannot grow normally anymore. So this happened in Texas a few years ago. So we don't want it happen in our rice field in Florida. So what kind of plant hoppers are present in the field in Florida? So we still have some plant hoppers, but they are not considered as pests. So we don't have any management practice targeting these plant hoppers. But one of the plant hoppers, we have it here. It's called rice delphacid. Togasodas oryzicolus. So this, based on Dr. Julian Buzelin's lab, they found 126 individuals during their survey in 2019 to 2021. So this species now it's considered established in the Florida rice field. And what is important thing to know is that this vector, this can act as a vector for rice Hoja Blanca virus. So they can directly feeding on rice, and then when the population is high, they can cause hopper burn, which is a huge problem. And if we have the virus present, they can also transmit the virus. Then it will cause bigger problems in the field. So you can see this is a poster. I got it from the Colombia. So this virus is in Latin America countries. They're causing problems. So this rice delphacid, they can transmit Hoja Blanca virus, and the virus infected with the rice infected with the virus will show these symptoms. And this virus, rice Hoja Blanca virus, it was reported in Mississippi, Florida, and Louisiana states in 1950s, 60s, and 80s. But since then, there's no report showing it's present, but we still need to keep an eye out, and then in case it's becoming a problem. And as I mentioned, this plant hopper caused the problems in Texas because the high population level, and it caused hopper burn. So what we are doing in our rice field is we keep monitoring plant hoppers number and then see if they're becoming an emerging pest, so we can have a management practice ready. And also we are testing this insect to see if they harbor any of these viruses. And once we find them, then we can let growers know before it's causing a bigger problem in the field. So for this one, Amir is going to tell you a little more about this.
Good morning, everyone. I am currently in my 7th year working here at IFAS. Most of it has been under entomology. I spent 1 year with Dr. Calvin Odero. My contribution to Dr. De-Fen's department consists of me and my colleague here, Tennyson, going out to 3 different locations in the EAA. Within those locations, we have selected 3 different fields where we take samples from. Once we gather up the insects, we bring them back to the lab. We put them in a -80 freezer. Once they've been sitting there for a good time and they're all frozen, we bring them out. I take them to the lab and I identify them. I put them in different family levels. And once I'm done doing that, I send them down to FDAX and they are further analyzed there.
Thank you.
Thank you.
Thank you, Amir. Yeah. Yeah, so so far those 3 projects are ongoing in the lab. So we have lab members involved in. And this project, the first one is in the pipeline. So we will start this project soon, but I just want to show you what we're going to do. So for this one is sugarcane aphids and sugarcane yellow leaf virus system. So for this one, so far we only had 2 or like one of the components in the disease triangle, but this one we have everything in Florida. So this will be our kind of exciting project. So here, as you know, sugarcane is the biggest crop in this area. We are the biggest sugarcane growing states in United States, and aphids is a common pest in sugarcane. And what they can do is they can transmit sugarcane yellow leaf virus. But the problem here is we don't know which species is exactly transmitting this virus. So just to give you some introduction, these 2 are the common aphid species in sugarcane. One is the sugarcane aphids you see on the left-hand side. So sugarcane aphids is a big pest, not only in sugarcane, but also in sorghum. So it's getting more attention nowadays. So you can identify them by their dark tips of their feet and antenna. And this one, the right-hand side, it's yellow sugarcane aphids. So they look very different from each other. So you'll be able to distinguish them easily in the field. So yellow sugarcane aphid, they have all the hairs covering their body. Both of them, they feed on sugarcane. So when the population level is high, again, they can cause direct damages to sugarcane because they uptake these nutrients from sugarcane. but the population level now, it's not too big to becoming a concern, but sometimes here and there, they are causing some problems. So growers, they are managing them by spraying insecticide. And our bigger concern is the virus they can transmit. So sugarcane yellow leaf virus is a common disease in a lot of sugarcane growing area worldwide. It's causing bigger problems in some countries. So in Everglades agriculture area, there was study conducted in 2018. They tested 204 sugarcane samples, and then 150 tested positive. So the virus is present in the area, and it's not a rare incidence. So, and you can see these symptoms. Once it's infected with sugarcane yellow leaf virus, then sugarcane will show this type of symptoms. And as I mentioned, sugarcane aphids, they are historically known for a pest of in sugarcane, but since 2013, there was a huge outbreak in Texas in sorghum. And this led to a study. They realized that sugarcane aphids, they have different genotypes. So genotypes feed on sugarcane and genotype feed on the sorghum. They're actually different genotypes. So they were speculating that they might have been several different invasions. So all of these are making this system more complicated because in Florida sugarcane field, we also have different genotypes. So a study conducted in 2018, they found that there are 3 different genotypes of sugarcane aphids in this area. You can see here H1, H2, and H3. So all of them were collected from sugarcane. So, but majority of them were H3 genotypes. And they can also infect the Columbus grass, and they can also feed on Columbus grass and sorghums. So they might have different preference. So if different genotypes of sugarcane aphids might have different preference towards their host plants. And this leads to our study project, which is to identify the vectors of sugarcane yellow leaf virus. As I mentioned, we don't know which species exactly transmitting the virus because sugarcane aphids is known as the vector worldwide. But the study conducted in this area in 2018, they had failed transmission using these sugarcane aphids collected in this area. So what I am guessing is that maybe the genotype, they have different ability to transmit the virus. Because this study they conducted in 2018, they only used the H3 genotypes, but not H1 and H2. And now, as we know, we have 3 different sugarcane genotypes in the area, and worldwide, they have discovered 6 genotypes. So what we are going to do here, it will be assess the genetic diversity of sugarcane aphids in Everglades agriculture area first to see how many genotypes we have in this area. And we are going to use different genotypes of sugarcane aphids to determine their vector competency. So my hypothesis is that although the H3 is the major, the sugarcane genotype, a sugarcane aphid genotype we can found in this area, but maybe H1 or H2, although they are minor, but they might have the higher efficiency in transmitting the virus. So that's why the virus incident, it's not like huge, but compared to the population level of the sugarcane aphids. And also we want to see what is their interaction. So different aphids genotypes and the host plant interactions. Because if you remember, you see from here, the majority of genotype we found on sugarcane is H3, and H1 and H1. So on Columbus and sorghum, all of them are H1 genotypes. So I think there is also some genotype specific interactions between aphids and host plants. So using this knowledge, we can develop something to see if we can use this knowledge to protect our sugarcane crops. And develop some cultivars. They are not preferred by these sugarcane genotypes, something like that. So there is potential, so research potentials here. And then this will be the major project for my PhD student who is coming in this fall. So please stay tuned. And so for the summary, now we presented our ongoing projects, preventing and protect Florida lettuce from INSV outbreaks and mitigate the impacts of a new invasive thrips parvispinus. And we are also monitoring our Florida rice field from exotic plant hoppers and pathogens. And in the pipeline is to study the interactions between sugarcane aphids, and sugarcane yellow leaf virus to understand this system better. With that, oh no, sorry, there is one more. So, what we are doing is some basic research. We are studying the insect ecology and the plant resistance to insects, vector identification, and then transmission biology. Also, we are doing applied research, field population monitoring, and insecticide efficacy. So we are combining all of this to help growers solve insects and vector problems and contribute to sustainable agriculture. With that, I would like to thank all the growers and crop consultant, these stakeholders. They are super supportive. So I'm very grateful. And the funding sources, also, I have established many collaborators since I joined here. So of course, I'm working with Dr. Julian Buzlin, another entomologist in here. And I'm also working with lettuce breeding lab and their lab members. And I also work with Dr. Hugh Smith and Alexandra Ravini in other research center, and also the extension agents here. They are super supportive about my project, and they've been very helpful. And also we are working with FDEC, DPI, and University of Georgia, and USDA ARS personnel. So I'm very grateful for these collaborations. With that, I would like to thank my lab members. Thank you. Any questions? I'm happy to take any questions.
Thank you for the presentation. That was excellent. I got to learn a lot about what your lab does, especially coming from soils and water, and you don't work with insects at all. But I just find it curious, why is it that they're the blue and the yellow pans, why are they attracted to specifically those colors?
Yeah, so usually yellow is commonly used as traps because they're kind of mimicking the flower colors. But then some of the studies that tested blues and also different colors, and then thrips seems like very attracted to blue colors. We don't know, maybe it's they're like reflecting, or like just the color different, because so insects use visual cues to identify and then to see which one maybe it's their host, right? They have to distinguish it's plant or this is soil, it's not their food. So something like something related to their behaviors and then their visual cues. Yeah, so we are trying to use different colors, but so far I think, so for sticky trap, I think blue is better, but then for pen trap, yellow is better. So we don't know like exactly what's going on, but that's what we see in the field. Yeah.
Just to add on that, normally, well, just as Dr. De-Fen said that yellow is related to the color of the flowers, but the color also of the sky is blue. So the blue color can also be related to that, because insects would think that they want to pass through the sky, and then they also get trapped into that.
De-Fen, I have a quick question.
Okay.
Hey, yeah, thank you so much, and your whole staff and lab, excellent presentation, and thank you all for presenting. Really enjoyed it. I was driving, so I got most of the seminar, but I wanted you to take a minute and just talk a little bit about the extension that you've been doing. How do you interact with the growers, and also do you involve your student and staff on those extension activities?
Yeah, thank you, Samira. So, growers been very supportive, and then since I joined, so with the help of extension agents, we get to visit the growers, and then now we are doing all this field work, like the insect survey in growers' field sites. So we have established the relationship with growers. So we visit them, and then so now we have this result we are going to also share with the growers. So let them know what's going on in their field. And if we find anything, then we can let them know as soon as possible. They can have better informed decision. And growers, they are also very supportive about our like ongoing research and then the proposal. So they also provide letters to support our grant applications. Yeah, so I think since I joined, I think we are doing pretty well with the growers. And then whenever we visit the growers, our lab members also join, and then so we get to introduce ourselves. So they know who we are, and then so they can find us like anytime if they need. Yeah, thank you.
Wonderful. Thank you, De-Fen.
Yes.
Just a question about the yellow leaf virus system. You talked about the diversity of the aphids. Is there much diversity in the yellow leaf virus itself, or is it pretty much all the same thing in South Florida?
Yeah, so I think there is one study from USDA, actually from Dr. Sushima's lab, but I think there are several different strains they discovered, but if I remember correctly, majority of them is from one strain. Yeah, but that's also something it's very interesting to look into. Maybe they have different like strain and then the genotype specific. So it's more like in molecular level, what's going on there. Yeah, thank you. Okay, yeah, so there is one, excuse me. Yeah, so there is one question from Zoom. So the question is, can you comment on approaches mitigating thrips parvispinus? Yeah, so thank you for the questions. So now this is a new invasive thrips. We don't really know too much about these thrips, but what we've learned from our once like season-long survey is that these thrips, they somehow have some different behaviors than other thrips. So from the plant samples, flowers, bottom leaf, and top leaf, we saw that from the flowers, we see a lot of other species of thrips, like Western flower thrips or thrips like Florida flower thrips. But these parvispinus, they are more in the bottom leaf. So that's something, it's very interesting because usually flower is the key samples that we take from the field. But if we only take flower samples, then we might miss them. They're hiding under the bottom leaves. But we don't know if this is because growers' application, so they are more like hiding in this area, or it's just their behavior. So what we are going to is continue using these different samples to see what's their behavior and what's the trend in the field. And then we are doing the efficacy trial I mentioned briefly, work with Dr. Julian Buzlin and Anna doing some efficacy trial. But so far, we see some effects on some of the applications, but we don't really have the definitive the recommendations for the growers yet. But in upcoming season, we'll still working together and working with growers to develop some recommendations to growers. And then we are going to also test some in the lab so we can combine this all knowledge and then present to the growers. Thank you.
Well, I had a question for Tennyson, but it's very focused on one little detail. In your non-choice assays, you infested your plants with 7 thrips. And I was wondering why not 8 or 6? And so how did you choose your number of thrips?
Okay, so I did some preliminary test to see which is a good preparation to use for that experiment. So I tested 5, I tested 10, I tested 6, I tested 7. So um, on there are two reasons why I choose 7 because it's a good number. Actually, when there's some kind of damage that can also happen in the process of taking the flips into the cage. If one flip is damaged, at least I can have a good number that is also surviving to feed on the plants. And the other reason is that I have a colony of thrips. So if I put a lot of thrips in just one cage, I might not have like enough population that I can use to infest the next batch of experiments. Yeah. And 7 is also giving a lot of multiplication in the code that actually used for preliminary experiment to understand the methodology.
And I just want to say something because I worked a lot with De-Fen and his crew and her crew. And I don't know if you haven't worked with trips. That's a lot of work. That's a lot of work as far as behind the microscope and sorting those traps out. And I know Yavon has been doing a wonderful job, and I was working a lot with Laura, and then that's a tedious work. And then we've been out there every single week from the season started back in August and taking weekly samples of those leaves and pen traps, and that's just so much work. And then what they just did and presented, it's just a little portion of it, but they were doing an awesome job. So just wanted to mention that.
Thank you again. I have a question. So I think it was in Tennyson's project where you had 10 or 20 different cultivars of lettuce, and then you would have the thrips choose which one. And you said it was because of perhaps the plant is excreting some type of biological molecule that is attracting those insects. Is that same biological molecule like used specifically for thrips? Like that wouldn't make sense. It would be more for like a pollinator or something like that. Like why are they being attracted to that molecule?
So for the choice AC, the main thing that I'm looking at is how attractive the plant is to the insect. So the attraction can also be related to leaf coloration or maybe other physical characteristics, like, for example, if the plant has some hairs, a lot of hairs, that if the insect want to feed on it, it will not reach to the plant tissue. That means the insect will not be going to that plant, and it will have to choose the plant that has little hair or the plant that has a good coloration that is actually attracting that insect to feed on it. So it's not related much to the pollination, but it's just related much to how attractive the plant is to the insect. So if I give them like the option to choose, uh, they will they will be on the middle, but they will go to the plant that is looking more attractive, and that makes that plant more vulnerable than the other plant.
Um, oh, yeah, if there's no more question, then I would like to thank my lab members again. Thank you very much for joining.